Can you hear me clearly? Okay, here we go. So far, I think we've been mostly talking about single-cell RNA-seq, and now we're going to switch gear. So the plan for my session, first off, I'm going to talk about some single-cell epigenomics, and more specifically, single-cell ATAC-seq, and followed by how do you do the alignment between single-cell RNA-seq and single-cell ATAC-seq data. So that's the first half of my session. The second half of the session, I'm going to be focused on cancer genomics. That is, we're going to talk about how you can use single-cell RNA-seq to infer some genetic profiles, for example, mutations in cancer. So the focus will be on cancer genomics in the second half. But before we go into the materials, I want to do a survey so that I know how much in depth I should go into. How many of you have worked on epigenomics or single-cell epigenomics? Please show your hand. Okay, great. How many of you have worked on cancer genetics or genomics? Okay, even more, great. Nevertheless, for the people who don't really work on these two different areas, I'm going to give you sort of, high level, but try to be clear as well, introduction, before we go into more technical details. Okay, and then like the first two speakers, I'm going to save all the questions towards the end of the presentation. Okay, so I'm going to start with single-cell ATAC-seq. So here's an overview for ATAC-seq. Basically, the DNA within a cell is not, fully stretched. You have 3 billion base pairs. As a matter of fact, they are actually, surrounding a structure that is called the nucleosome. So in certain regions of the DNA genome, the nucleosome are quite dense, as you can see here, whereas in other regions of the genome, the nucleosome seems to, there are some stretches in between the two different nucleosomes. So, if it is sort of like this piece of the DNA, as I show here, we call this the open chromatin regions. So the ATAC-seq basically is trying to infer which region of the genome actually is open. So it assesses the open chromatin regions. Okay, and then it does this in a high throughput manner. So more details, basically, during the library prep step, basically you, insert this TN5 transposes, and it can actually insert sequence adapters to the open chromatin regions. And then followed by this, you'll basically, cut this off and then do the sequencing. Okay, and then when you look at the sequencing, the read length, after you do the cuttings, usually you see this sort of like a rhythm pattern in this case. And the reason is that you basically, you cut in between two different nucleosomes, and then, basically the distance between each individual peak here is the length of the nucleosome. So if it, goes through the loops of one nucleosome, you will see, the length pretty much of one nucleosome, so on and so forth. Okay, so this is actually the QC step, during the pre-processing of the ATAC-seq. When you see a very nice sort of like a rhythmic pattern here, you know that your data quality is pretty good, and you go from here. Okay, so why do we need a single-cell ATAC-seq? Why can't we just, adopt the traditional bulk ATAC-seq? I guess the same thing goes for single-cell RNA-seq as well. Shown here is the, cartoon, basically in this human hematopoietic hierarchies. You see that for all these different types of human blood cells, you have these different subtypes of cells. So, as you can imagine, if you sequence a bulk ATAC, it will just be, all averaged together. Now, obviously, the single-cell ATAC-seq will give you a much higher resolution in this case. And then on this right-hand side, basically what I showed on the, I think on the top, all of these are actually bulk sequencing. So this is a bulk RNA sequencing, this is a bulk ATAC sequencing. But all of these samples are actually collected after you do effect sorting. In other words, all of these bulk samples, even if they are bulk sequenced, they're still pure cell types. And you can see that they actually cluster by their respective class rings. And more importantly, you see that by RNA-seq and by ATAC-seq, they seem to share the same cluster patterns, between these two different types of measurements. Okay, so this actually leads to, it kind of motivates us, try to align these two data sets to increase your power for identifying the cluster memberships. Okay, obviously you see here they share some resemblance, but they are not exactly the same. In some cases, for example, the single-cell ATAC-seq, the ATAC-seq might actually give you, a clearer separations of the different subclusters. Okay. So in terms of the specific protocols for single-cell ATAC-seq, I would say that so far there are two main streams. So the first one is sort of like the FluDyme version for single-cell RNA-seq, where you basically isolate each of the single cell into its specific chamber in your 96-well plate, and then you do all the reaction within that chamber. So you sequence each individual cell individually. So this combinatorial indexing, well, I guess on the right-hand side, it's a FluDyme. So on the left-hand side, it's the combinatorial indexing protocol developed by Jay Shunder. It has been used for both single-cell RNA-seq and single-cell ATAC-seq. And I think recently they published a paper that they use the same strategy to profile single-cell RNA-seq and single-cell ATAC-seq simultaneously from the same cell. But nevertheless, for this technology, basically the idea is that you don't really individually separate the cells, but you sort the cells through maybe 96-well plate several different times. As you can imagine, each of the cell, as they go through different steps of barcoding, the probability of two different cells share the same set of barcode will be very small. It's sort of like you can neglect this artifact that the two cells after rounds of barcoding will share the same set of barcode. So as you can imagine, after the final sorting, you can basically each of the individual cells will be barcoded with a unique barcode to that specific cells. Does that make sense? Yeah, okay, so and after this, basically you can just pull all the cells, do the Illumina sequencing, and then based on the barcode, you know which read correspond to which cell. So these are the two mainstream techniques for single-cell ATAC-seq, and here I think it's just to show you, compare to single-cell RNA-seq, as well as the comparison between bulk ATAC-seq and single-cell ATAC-seq. What is the sequencing depth? Obviously, the single-cell ATAC-seq coverage is way lower compared to your traditional bulk ATAC-seq. It's also way lower compared to your RNA-seq. And then between the two different techniques that I told you, the combinatorial indexing has very low sequencing depths per cell as well, whereas FluDyme C1-based techniques, it can do sequence much higher. And then here is just a visualization. Basically, you see that for the single-cell ATAC data compared to the bulk, the sequencing depth is significantly lower. So there is always a trade-off between the sequencing depths as well as the resolution or the number of cells that you can sequence. So for example, for the combinatorial indexing techniques, we can easily sequence thousands, hundreds of thousands of cells. However, for the FluDyme C1, usually you plot the cells into a 96-well plate. Some of them may fail, so you have around 90 cells to be sequenced. I guess I can probably use this. Yeah, so here is just a detailed visualization to zoom in into a specific region on chromosome 1. Okay, so all of the cells or samples that I showed you here are of the same cell type. And then I think on the bottom part, it is a purified bulk samples. And when you see these peaks here, basically it means that you have an open chromatin accessibility region. And then for the single cells, again, they are of the same cell type as the purified bulk samples, but you see here that it's highly sparse. You have tons of zeros in this case. To be more exact, in single-cell ATAC-seq data, oftentimes you only have around 1% or even less than that reads as non-zeros. And I think I have a table here to show you the percentages of zeros and percentages of ones are greater than or equal to 2 in the single-cell ATAC-seq data. You see here, even by the FluDyme C1 techniques, out of all the peaks that we profiled, around 97% of those are actually 0. Another thing I want to point out here is that the dimension here is way higher than the single-cell RNA-seq as well. So the single-cell RNA-seq of your QC, you probably have 15,000 genes, whereas in here, for single-cell ATAC-seq, the dimension is hundreds of thousands of peaks, to begin with. So the dimension in this case will be, almost 200,000 peaks by 1,000 cells to begin with. So given this fact, a lot of the techniques that we have developed for single-cell RNA-seq data cannot be directly applied to the single-cell ATAC data. The first reason is that the data is just way too sparse. You barely see any reads, and then even if you see read, it's most likely going to be 1. And then secondly, the dimension is just way too high for even the PCA to run properly. Okay, so how do we deal with this issue? A few methods have been developed. The first method, scRAT, from Hong Kai-Ji's group at Johns Hopkins. Basically, they try to aggregate the different peaks that I talked about into the same biological annotation regions. So basically, you can either try to combine all the peaks that fall within the transcription factor binding sites for the same transcription factor. You can also have a very large region of the genome, say 500 kb, and then you bin all the peaks within these 500 kb regions. And you can also basically bin all the peaks within a gene or the gene of the gene, the same gene set enriched. So basically, the gene sets that are interested in. Okay, so basically the main goal for this step is to reduce the dimension so it's computationally feasible. Okay, so and then this will be followed by a further dimension reduction, to further reduce the dimension here. And then basically, I think what they did is just a PCA, based on the aggregated, matrix, and then based on the PCA, they will do the clustering. Okay, so what do you think the problem can arise from here? And I think I probably have a more specific explanations in the next few slides, but just to elucidate a little bit here. So basically, how you actually aggregate all the peaks in these steps really depends on the cell types. How you do the cluster of the cell types you're interested in. Okay, so basically you want to cluster the transcription factors, and then the transcription factors better to be your cell type cluster specific. So it's like a cyclic problem. So where do you start? Do you start with clustering first to infer the TF binding, or do you start with TF binding to infer the cluster? It's like a chicken and egg problem. To us, it feels like we need to be more careful with regard to this method. Okay. So this is another method developed by Wing Wang's lab at Stanford. It's published in Nature Communications last year. Basically, they try to adopt a model-based clustering method. And then in this case, I think the unique property for this method is that they take into consideration how deep the cell is sequenced. So if the cell is sequenced at a higher depth, it will give higher weights when you do the clustering or when you do the modeling. If it is sequenced at a lower depth, vice versa, you will give it a lower weight. Okay, so I skipped a lot of the details, but basically it's a model-based clustering techniques. And then the clustering, after you do the calculations of the distance of the cells, you basically adopt a k-medoids clustering methods that can give you the clustering memberships of all the cells. Okay, I want to point out one more thing. I talked to a few of you during the break. Basically, I got the feedback why you don't really spend time on clustering, because that's usually what we are really interested in. So I think there are two reasons why we do this. The first reason is that we think the clustering is usually the second step. The first step will always be how do you normalize your data? How do you do denoising? How do you do imputation? So on and so forth. So we feel that the first step is the most important step. And then after that, you can adopt the various different techniques based on the imputed, normalized, corrected, or whatever matrices you get. Okay, so that's why we don't specifically focus on clustering per se. And the second reason, there are a few clustering methods and benchmark papers that we put on the GitHub page. We feel that it's relatively easy to follow, and then feel free to check on those, at your own time after this session. Okay, so that's sort of a detour, but going back to here, this is a method that we developed from my lab. So instead of trying to aggregate the peaks based on pre-annotations, we don't do any aggregation first. We just throw all of the peaks into the clustering step. However, here, we also try to put a weight. So the weight will just be based on the distance to the TSS sites, as well as the frequencies of the open chromatin accessibility regions based on previous studies. That is an ENCODE project that we adopted. And then, this is a benchmark that we carried out in our method. In our paper, it seems that it performs really well. And in a recent paper, I think published a few weeks ago, where they benchmarked the various different techniques, it seems that our method performs the second best. Well, the best one obviously is the one that they propose in their paper. So we feel like here that we're quite confident that this scheme, even though very different from the mainstream of analyses nowadays, it seems to work really well. Okay. I've talked about how you can do the clustering by single-cell ATAC-seq itself, and then here we'll move on to how do you do the alignment between single-cell RNA-seq and single-cell ATAC-seq. Before we talk about this, you may wonder, why don't you just adopt the techniques to sequence both the ATAC and the RNA from the same cell? Well, there are technologies that are actually available. However, first off, it's very expensive. It's not very easy to implement. And more importantly, we've actually looked at the data set where they sequence, the two modes of data from the same cell. The single-cell ATAC-seq, you see even more zeros. So for a typical cell that is sequenced, bimodally, you will get a few hundred reads for the ATAC only. So we feel that it's just way too sparse for you to do anything further. So anyhow, basically, what I want to say here is that still it is very important for us to develop some methods that are rigorous to, align these two different types of data together. Okay, so this is a illustration here. So basically, it's all of these cells are PBMC cells. On the left-hand side, this is a t-SNE visualization for the single-cell ATAC-seq. On the right-hand side, this is a t-SNE visualization for the 10x 5' tagging single-cell RNA-seq. And I haven't shown you, there's another version of the 3' tagging for 10x as well. But basically, you see here that the general structures do seem to resemble between the single-cell ATAC-seq and the single-cell RNA-seq. And furthermore, if you do the proportion calculations of all the different cell types, they seem to be fairly similar to each other as well. So in other words, the different cell type information seem to be preserved between the single-cell ATAC-seq data and the single-cell RNA-seq data. So in other words, it might be feasible for us to computationally align these two different data sets, even though they are not generated in the same exact cells. So here is just another view, but basically we highlight the transcription factor binding for the ATAC-seq. So basically the peak for the transcription factor binding sites for the ATAC-seq data, or the gene expression for this transcription factor in the single-cell ATAC-seq data. Okay, so you see here again, they seem to show fairly similar pattern with some difference, obviously. So basically, the notion here is that it seems that the more open the region is, the more transcription factor binding there is, the higher the gene expression you would expect in this case. And here is just an even more complicated situation. Not only do you have two different modes of measurements from the cells, but also you have a case control status. So on the top panel, it is ATAC-seq, this is RNA-seq. And then, for each of these measurements, you have a healthy and you have I think it's a leukemia patient, the cells from the cancers as well. So, just bear this in mind, it can get complicated very easily. Okay, but nevertheless, still you see that the structure seems to be, shared between the different cell types by different technologies. Okay, so how do we do this? This paper based on coupled NMF from Wing Wang's lab, we feel that it's the first method that tried to tackle this issue. It is published in PNAS last year. There is some issues to be resolved, but we still think that we should give them credit in the sense that they are trying to first propose this problem and offer a solution to it. Okay, so it is based on coupled non-negative matrix factorization. And then, basically they adopt a soft clustering. So how does this work? Really briefly, NMF, if you apply this to single-cell RNA-seq itself, basically you want to decompose the observed gene expression O, which is the number of genes by the number of cells, into the product of two non-active matrices. So the W1 will just be the number of genes by the cell clusters. The H1 will just be the clusters by the cells, okay? So the first one, the W1, you can think of it as your profile matrix for each of the cell clusters, and then the H1 will be your cluster memberships for each of the cells to each of the cluster. And the reason that they say this is a soft clustering is because they don't do the cluster deterministically, but rather through the H1 matrices, it's a proportions where all the columns actually add up to 1. So with the proportions being the probability of you assign a specific cell to a specific cluster. Okay, so you can do this for the gene expression matrix. That is, you do that on itself. You can obviously do this for the ATAC-seq matrix as well. So the only difference here is that instead of looking at genes, you'll probably look at the peaks, or you'll look at the transcription factor binding sites by the number of cells. Okay, so we also call this as the alignment problem. However, I feel that this further complicates the problem in the sense that not only do you not share the same set of cells or samples, but also the features. So the number of features differ. For the gene expressions, it's the number of genes. For the ATAC-seq, it's the number of peaks or the number of loci. Okay, so the dimensions is completely different. Whereas for when Jingshu talk about when you try to align CCA, at least the number of genes will be shared between the two single-cell RNA-seq dataset. Okay, so you can do the NMF for the RNA-seq, for the ATAC-seq. You can add some penalization here. And then the most interesting part is actually this part here. Basically, they call this a coupling matrix. Okay, so basically, remember we're talking about this W2 matrix and the W1 matrix. And then the A matrix will just be of the dimension P1 by P2. So the P1 will be the number of features in your first measurement, or I guess in this case, maybe P2 by P1. So nevertheless, it's of the dimension, the number of features from the first set versus the second set. So it tells you basically, how you can predict from the gene expression to an ATAC-seq or vice versa. So basically, it tells you the association relationships between the ATAC-seq and the RNA-seq dataset. And then this A is actually a known input for this method. And how do they get the A? They get the A from the ENCODE data, where they have the purified cell lines, and then they have both the RNA, and you have the ATAC-seq. And then you can learn from that sort of as a training set to learn the relationships between these two. Okay, so like I said before, the construction of A is application specific, but also it depends on the assumption that we're trying to identify a subset of features in one sample that are linearly predictable of the features in another sample, okay? And then here is what I previously talked about very briefly. There is a problem of circularity. Basically, where do you start? It seems that the prior data very heavily depends on your true cluster memberships and vice versa. Okay, so there's another method published in Genome Biology. So this method also tries to align the two different data sets at the single cell level. It's called Matcher. So basically, how it works for this method is that they try to infer the pseudo time or the trajectories for each of the data separately. So you infer this manifold by gene expression, you infer the manifold by chromatin accessibility, and then you try to merge these two manifolds together, sort of in an ad hoc way. Okay, and then it seems to work for some cases. However, we feel that the substantial drawback for this method is that it doesn't really allow any branching in either data set. So basically, the assumption that the cells actually lie along a one-dimensional manifold might not always hold in real data set analyses. Okay, so I think just a few months back, there's another method published from Rahul Sathija's lab, and then that again is based on CCA. So here is just a few more details about this method. So they began talking about the CCA by merging two single-cell RNA-seq from two different batches. But the same notion applies here. Basically, you can think of instead of aligning two different single-cell reference set, you're trying to align the ATAC-seq and then the RNA-seq. Okay, so what they do here basically is they want to find a shared gene correlation structure that is conserved between the two different data sets by applying CCA. This will be after applying the CCA. I have one more slide to show how exactly this works. But after you do this, you see here that clearly the cells don't share the same CCs between the two different batches. So based on the CC1 and CC2, you have to correct this so that the cells from the different batches can be well aligned. So how do they do that? They do this in a step that they call the dynamic time warping step. Basically, after this, the CC1 from the two different batches will be well aligned. They call this ACC1 and ACC2, for example. We feel that this may seem a little bit ad hoc, but it seems that for certain data sets it works really well. Okay, so after they do the alignment, they can do some integrated analyses. For example, you can tell what is the proportions of the different cell types between case control status. You can do a differential testing in this case. And then for a specific cell type, you can plot the gene expression profile for one condition versus the other condition. And then you are interested in these off-diagonal genes. And then I guess this is a slide that I mentioned about the CCA. Basically, instead of PCA, where you try to find a linear projection that maximizes the variance after you do the projection, in this case, you're trying to find two different vectors that maximizes the correlations that are shared between the two different data sets. So this is another visualization, but I think I'm going to skip. So this one is a CC1 before correction. You see here that between the two different data sets they want to align, obviously the CC1 not only capture the biological information, but also seems to capture part of the batch effect as well. But if you do the alignment, it seems that the batch effect is correctly taken care of. Okay, so this method is published in Cell. And then they have a specific demonstration where they analyze the RNA-seq and then the ATAC-seq together. So this is a t-SNE visualization after the alignment. And then this one basically is the same as here, but they color the different cells according to their cluster memberships. And then the backbone of this method is that basically they use Monaco 2, I believe, trying to predict the gene expression from single-cell ATAC-seq data. So in other words, it will have a pseudo gene expression data to align with the actual gene expression data. Does that make sense? So basically, they're trying to predict a single-cell RNA-seq data from the single-cell ATAC-seq using Monaco, and then align the two different single-cell RNA-seq data together. Okay, however, the problem here is that the single-cell ATAC-seq might be too sparse for you to do the prediction very accurately. And then there's another method that try to do in a reverse way. Basically, they're trying to predict the single-cell ATAC-seq from the single-cell RNA-seq, and then align the two different single-cell ATAC-seq data. And then the alignment that they use here is the MNN, which Jingshu talked about as well. Okay, so it's like two different ways how you can deal with this issue. But nevertheless, I think for either method, they're trying to first transform all the data into the same dimension, either ATAC or RNA-seq, and then do the alignment. Okay, so these are the references, and I think this is what I have for the first part. I'm open to questions. Or if not, I will just move on to, okay, yeah, go ahead.
Just a fundamental question. What do you really expect to gain by combining these two data types? Because it seems that chromatin accessibility is a necessary but not sufficient condition for gene expression. But if you already have the gene expression, you're already a step further. What can you expect to to gain if you also have chromatin accessibility?
But yeah, I think that's a very, very good question. Why do you do this to begin with? And I think that might be a question that is, better answered by a biologist. I guess maybe you can refer to the Cell paper. What they did following upon the alignment. I think they look at some of the, transcription factors, some network analyses, so on and so forth. But that is further downstream. But again, I think this is a very good question. Yeah. Any more questions for this? Okay. All right, so now we're going to switch gear to talk about cancer genomics. I know the only thing between you and lunch is me, and a lot of you are very jet lagged, like I am right now. So I will try to emphasize a part that I feel interesting and important, and also I will try to be fast so that we can all, get up early. So cancer genomics. Here is just an overview for those of you who really work on cancer genomics. I guess this is probably just a review. We know for decades that the cancer samples are highly heterogeneous. And then shown here basically is a cartoon to illustrate this. Start with normal cells. Sometime along the cell division, a very bad exogenous shock happened. You mutate it into a cancer cell, and then we call this usually the founder of all the cancer cells. And the cancer cells, presumably, they can divide and divide infinitely. And then as the cancer cells divide, some additional mutations can be acquired. So this will lead to a new branch here, colored in a different color. And then we call these different colored cells the different clones for a specific cancer. And then, at a certain time point during the tumor progression, it's diagnosed. So you found the primary tumor, so you apply some bottleneck, you apply some treatment, as a selection bottleneck for all the cancer cells, okay? So, for example, the chemotherapy works really well in killing all these cells, but the sneaky green cell somehow, it survives this selection bottleneck, and it leads to the metastases. And unfortunately, the outcomes for all these metastasized cancers are usually very poor. Okay, so the take-home message over here is that you have all these different types of subclonal dynamics, either between different time points or between different locations of the tumor, and more importantly, for a given tumor, it is highly heterogeneous with multiple different subpopulations. So if you do a traditional bulk sequencing, especially if you only do it once, you will just get a very blurred picture or snapshot of what's really going on within the tumor samples. Okay, so this is why we think single cell can be especially powerful in cancer genomics, in the sense you can actually zoom into one specific cell and study what's really going on in that cell. Nevertheless, between the single-cell era, a lot of efforts have been devoted to either the science side or the method development side, trying to tackle this issue. Basically, we want to infer the evolutionary trajectory even without single-cell sequencing. So usually what has been done is that we take the bulk DNA sequencing, but we don't do it once. We do it multiple different times, either at different time points or at different locations. In other words, you want to take a snapshot, but don't take it at one angle. You want to switch your angle, so hopefully you can get a more complete picture of what's really going on in the tumor. Okay, so in this sense, the study designs fall within either spatially separated tumor samples or temporally separated tumor samples. So for each of these cases, you collect multiple different bulk samples and you sequence this by bulk DNA sequencing. And then we have developed a method called Canopy. I'm not going to talk about these details, but basically this is using bulk DNA sequencing only, and then we're trying to infer both the point mutations and the copy number variations, and how these two types of mutations work together, and then how we can use these two simultaneously to infer the evolutionary trajectory of that specific tumor that you're interested in. Okay. Again, I'm not going to talk about these details, but the paper is here. The method is in GitHub. So now we move on to single cells. That's what everybody is talking about. Instead of looking at a population of cells, now we can pick out one cell and then we do the single cell sequencing. As a matter of fact, there are a couple of studies have already adopted single cell RNA sequencing to study cancer. And then these are 3 Nature and Science papers published from the Broad Institute, where they specifically study the gliomas, okay? So there are 3 subtypes of gliomas, IDH A mutant, IDH O mutant, and it seems that this is an IDH wild-type glioblastoma. So in these 3 papers, they study these 3 subtypes of gliomas by single-cell RNA sequencing, respectively. And here I'll just very briefly summarize what they find in general from these 3 papers. So what they do is that they sort of do some dimension reductions. I'll skip the details, but these are sort of like the PC1 associated genes. These are the PC2 and PC3 associated genes. And then basically they find that the genes that are associated with PC1 seems to be highly expressed or lowly expressed in the prenatal stage, and then vice versa. As you age, it seems that the PC2 genes and the PC1 genes seem to have different patterns. So in other words, their conclusion here is that the PC1 are associated with the lineage. So they call this a lineage score, and the PC2 and PC3 seems to represent the stemness signatures. They look at some of the gene signatures for stemness, so they call this a stemness score. So they basically draw this out, and this will be the trajectories, and then if you go up here, it will be more stem-like. If you go down here, it will be more differentiated, and then this one will be further differentiation into astro-like or oligo-like cells in this case. So each point here is one cell, and then they reconstruct this structure purely based on single-cell RNA-seq data. Okay. So one thing that they briefly mentioned towards the end of their paper is that they look at the genetic mutations of all these cells as well. Bear in mind that all of these is done based on single-cell RNA-seq only, but we know that the cancers are mostly separated not only by RNA expression, but also by their mutational profiles. Whereas in this case, when they do the analysis, they just completely ignore what the mutation profiles are for each of the individual cells. Okay, so towards the end of the paper, basically, it's the same structure, but they color the cells based on their copy number variations or based on their point mutations. And their conclusion is that the tumor architecture was preserved across clones reconstructed by CNVs or point mutations. In other words, it seems that all the mutations seem to span along all the trajectories that you see in this case. However, in their discussion section in multiple papers, they also point out that without a comprehensive phylogenetic reconstruction, we cannot categorically rule out a genetic influence. And in another paper, they say future studies should further investigate the modulation of our inferred cellular architecture. Again, this is by RNA only, by genetic evolution. In other words, as they pointed out, basically, the next step, it seems that you want to integrate both the genetic information as well as the transcriptomic information from the same cells. It's like genetics is one of your confounders. When you do the GWAS studies, you have to correct for the ethnicities for each of the individuals. In this case, the confounder is not your ethnicity, but it's your mutational profiles. Okay, so how do we infer the mutations to begin with? You can traditionally adopt a bulk or single-cell DNA sequencing to infer copy number variations. I guess this is the most intuitive way because you are sequencing the DNA directly. Single-cell RNA sequencing, at the same time, it can be used to infer copy number variations as well, although the resolution can be very low. And I will show you how this really works in a few examples. Point mutations for bulk DNA sequencing, a lot of methods have been developed, how you can call permutations, how you can do annotations. So I'll skip this. For single-cell RNA sequencing, I'll briefly mention what are the current issues and what are some of the available solutions. Okay, so we'll start with the first one, how you can infer the CNVs by bulk DNA sequencing or single-cell DNA sequencing. So CNVs, I guess for this audience, I can skip, but basically, it's very important. It's largely deletions and duplications, and the tumor can be very messed up in terms of their copy number profiles. And then this is one of the methods that we developed. Basically, it's a Poisson latent factor modeling. The reason that we adopt this, especially the exponential factors, these latent exponential factors, is because the next generation DNA sequencing is very noisy. So you need all these different factors corresponding to the different sources of artifacts and biases. So you want to capture all of these so that your CMV signals will be retained from the artifacts. Okay, I think I'm going to skip these details, but here is just a view how it works. You start with the raw coverage. You try to model all the artifacts, and then through the normalization step, you will find this normalized matrix. And you see here that in this region, we know there is a nested deletions here. This will be our positive control, I guess. And then after this, the normalization step will just perform the segmentation to attain a whole region that has a uniform copy number profile. This paper has been published for a few years. And then since then, we further develop another enhanced version we call CODEX2. So in this case, we show that with the utility of negative control samples, which is your normal samples in your cancer genomic studies, or your negative control regions. For example, these are the regions that has nothing to do with CNVs in my breast cancer study. So we show that with the utility of these two different terms, you can significantly increase your sensitivity for CNV detection, especially for common CNVs. So that paper is published in Genome Biology. And then here, just to give you a visualization, with the first version, you don't detect you nearly detect any of the duplications for BRAF or the deletions for P10 in this breast cancer cohort. Oh, no, wait, so for this melanoma cohort. Whereas for the CODEX2, with the utility of the normal sample as controls, you actually recover all the expected CNV signals in your cancer samples. Okay, so that is for bulk DNA sequencing. And then on top of all these, we recently developed SCOPE. Basically, this is for single-cell copy number estimation by single-cell DNA sequencing. The paper is available on bioRxiv. And here, I think I will just give you a very brief overview. So each of the row here is one single cell that is sequenced by single-cell DNA sequencing, and then each column is one chromosomes. So in this case, you can clearly see that you have one small subclone that is detected by single-cell DNA sequencing because of the resolution where it is completely missed by the traditional bulk DNA sequencing. I mean, on the ballpark, the whole exome sequencing, the profiles by that agrees, almost perfectly with what we find for the major clone. But usually, it is a rare cell subclone that we're interested in this case. Okay. So now we're going to move on to single-cell RNA-seq. Is this too fast? Or are we good? Okay, so for single-cell RNA-seq, this is a method that is developed, by I think Itay Tirosh, while he was still at Broad Institute. So basically, in this case, you have the assumption that the higher the copy number profile, copy number that you have is for a cell in a region, the higher the expression it will be. Vice versa, if you have a deletion, you will expect less expression from this region. But obviously, the gene expression is not only modulated by copy number only, but it is also, you have a lot of stochasticity for gene expressions. So in other words, in this case, you have to really zoom out into a very large region when you can have this pattern to show up. Okay, so this is actually, application results on two glioblastoma patients that I introduced before. And in this case, you can detect the chromosome or chromosome arm level deletion or duplication. But it seems that this is the best that we can go with the single-cell RNA-seq data. Okay, so the method is available here in GitHub. So how do you identify the normal malignant cells? So the method actually uses normal cells as a baseline to correct for all the gene fluctuations. So the first step is how you can actually identify the non-malignant cells. In this case, I think the consensus is that you do a dimension reduction first. In this case, each column here will just be one cell. Each row will be one PC. And then this color annotates the different sorts of the of the cells. And then you can see here on the very left-hand side, these group of cells seems to be coming from different patients. So these are actually the normal cells, which shows higher resemblance across patients. Whereas if you look at the cancer cells, they are, due to the intratumor heterogeneity, they most likely clustered by the patient source. Okay, so in this case, PCA can sometimes help us to pinpoint out which one is actually the normal cell, which one is the tumor cells. And then for the single-cell DNA-seq, we actually adopted the Gini coefficient. Basically, you can think of this as an assessment of how uniform the coverage is for specific cells. And in this case, you have a normal cells. It's sort of like a unimodal shape in this case, with a Gini coefficient very close to 0. Whereas if you have a tumor cells or you have an outlier cell that fails the library prep, the Gini coefficient will be significantly larger compared to a normal cells. So we feel that this can also be another way to quantify the normal cells out of all the cells. Okay, and then this actually is further validated by the effect sorting results. Basically here just to show that on the y-axis are the Gini coefficients. X-axis, you can think of this as what you get from effect sorting. And you see that, again, for the diploid cells, they have significantly lower Gini coefficients compared to the tumor cells. Okay, so finally, I'm going to talk about the existing methods that try to simultaneously model the genetic mutation profiles as well as the gene expression variations. There are two published, there's one published method from genome research. This one is still on bioRxiv. So the first one tries to tackle the issue on how you can better quantify copy number profiles based on single-cell RNA-seq data with some follow-up studies, which seem interesting. And the second one is focused on using the point mutations only. Okay, so the top one, both of these are for single-cell RNA-seq, but the top one is focused on copy number. Bottom one is focused on point mutations. So the honey badger, published by Peter Kachenko's lab, basically they try to use a computational approach to infer the deletions or duplications from the single cells. They did point out that there are existing technologies that are available that try to simultaneously infer DNA and RNA, but again, this can be very expensive, and the technology might not be as mature. So for single cell only, I think what they did in this case, very briefly, is that they look at the allele frequencies. Basically, you have a deletion. In this case, you'll most likely see almost 0% for the heterozygous allele in this case. Whereas if you have a neutral region, you will have randomly pick up one allele to be expressed. Okay, it's very similar to the array-based Simile error where you have a B allele frequencies. So if you have a 0% or 100% that is enriched, it will most likely be due to a deletion. Whereas if you see around 50%, it will be a neutral region, copy number neutral region. Okay, so they do this using a hidden Markov model. I think I'll skip this details. It's not very detailed anyway. So basically, based on the gene expression profiles, they adopt a hidden Markov model with the hidden states being the copy number state. Okay, so there are 3 different hidden states. In this case, it will be a neutral region, a deletion region, a duplication region in this case. And then shown here is just a transition probability matrix, for this hidden Markov model. However, we think that this, can be maybe further parameterized. For example, the T here is shared across all the chromosome, all the regions. Maybe you can add a few hyperparameters in the key, in the T here to further boost the performance. Okay, so this Cardellino is another method that I said they try to infer the point mutations from the single-cell RNA-seq. So the way that they do this is that for a specific tumor specimen, they first use the bulk DNA sequencing to infer the clonality structure. As a matter of fact, I think they adopted our method, Canopy, that I told you, the PNAS paper, to infer this clonal structure shown here. And then after they use a bulk DNA sequencing to achieve this structure, they do a probabilistic assignment of each of the single cell to each of the node in this case. Okay, and then after they assign all the cells into a specific node, they can do some further analyses. And then, basically all the analyses will be restrained to a specific clone. In this sense, you will control for all the genetic variations. Okay, but again, I may feel a little too cherry picking in this case, but we feel that this is not fully using the single cell information. In the sense, the clonal structure, it is still purely constructed by the bulk sequencing only. So how you can jointly infer this structure by both the bulk and the single cell DNA sequence, single cell RNA sequencing might be interesting to investigate further on. So again, as a summary, they first infer a clonal tree using varying allele frequencies from bulk or single cell DNA sequencing data, and then performs probabilistic assignment of individual cell transcriptomes to infer clones using varying information extracted from the single cell RNA-seq. And then there's actually another complicated issue for single cell RNA-seq. So basically, we know that the gene expression is very stochastic. It's not always constitutively expressed, especially when you look at the single cell level. So we call this a transcriptional bursting. Basically, if you look here, the genes can be on and off, and then when the gene is on, it will just burst out a few transcripts. And then by chance, when you do the single cell RNA sequencing, you are taking the snapshots in this case. In other words, if you are really unlucky, and then you take the snapshot here, you see 0, 0 from that cell, it doesn't really mean it's a true 0. It can sometimes mean that the transcript is not being expressed. So the cell that you see 0, 0 might still be your cancer cells, even if you don't see any mutations that is captured by single cell RNA-seq. To account for transcriptional bursting, there is this method. It's still on bioRxiv, but basically by single cell RNA sequencing, you can use whichever variant profiling method you like. It can give you the number of alleles that actually have the mutant reads, as well as the number of alleles have the total number of reads for a specific allele. Okay, so when calculate the distance matrix across all the cells, it takes into consideration the transcriptional bursting effect. Okay, so in other words, what I mentioned here is that the zeros that you see here that has 0 mutation profiles, it can be, the cells can still have that mutation, it's not being expressed. Okay, and then so in the distance calculation step, they try to integrate this part through a modeling step as well. And then in this case, we'll just come up with a cell-by-cell similarity matrix. And then they do one round of hierarchical clusterings, and then they combine all the cells from the same cluster, and then you do profiles of all the subclusters through the hierarchical clustering. Okay, so I think I will stop here. It seems a lot of these are not in great details, but again, we have all the list of the methods that we introduced on the GitHub page. We have both the link to the paper as well as to the software. Feel free to take a look after this. Any questions?
Nice talk. I have a question actually about copy number profiling. For example, this studies initial when that was done on the single cell RNA sequencing, right? For example, in the gliomas, it was this full transcript length, right? The, for example, C1. But in this 10x, right, and drop, we have only 3'. So what about this? Are there any methods which take this into account? Is this under development?
I think in this genome research paper, they claim that they can do something with the 10x data, but my understanding is not very, you probably only can infer the copy number change on the chromosome level by 10x data. Yeah. Any other questions?
Hi.
Yeah.
I have a question. So in terms of mutation detection, when you do single cell sequencing, compared to bulk. You mentioned also briefly that you can do it with RNA-seq, single cell RNA sequencing in the last slides. So do you know actually how, whether we see really differences in the mutations detected? Is it just about, so I just try to understand what the gain is in mutation detection when you do single cell. Do you see additional mutations that you don't see with bulk? Or is it just about, so that you can divide the mutations into different subclones?
Yeah, that's a very good question. I think, no, you don't detect additional mutations by single-cell data, especially by single-cell RNA-seq data, because most of those will be highly contaminated with false positives. You have a lot of RNA editing, you have a lot of sequencing errors. So most usually, we start with bulk DNA sequencing to pinpoint where the variants are, and then strictly call the coverage of the in the single cells at this specific loci. So this again goes to the question: why do you do all these again? So in this case, I think what they want to do is to use all these variants to assign the single cells into a specific clones. Otherwise, you don't know where the cells come from. It's just a whole bunch of cells. Yeah.
Yeah, just one comment and one question on the. So with single cell amplicon-based sequencing, you do detect a lot more mutations, the coverage. A lot of the droplet-based methods are there, like from Mission Bio and platforms. My question is, in the codex, does it detect? Does it estimate integer copy numbers, or does it estimate integer copy numbers, or just gain and loss for codex and codex 2?
Estimate what? Absolute?
Absolute, yeah.
Yeah, it does. It does. It's not only returning 3 states, but also the specific copy numbers.
Okay, it's on whole genome, and can it be also applied on amplicon-based?
Yeah, definitely. I think it's actually more useful for targeted sequencing. Okay. Yeah. All right, thank you, guys. Yeah. Oh, by the way, one last thing. Before all you go, please, or after you go back, please fill out this survey. We would love to hear back what you think. Okay, thank you.